Today we are discussing enzyme inhibition and control. Because all of the metabolic pathways in the cell are governed at some point by enzymes, the ability to inhibit and control enzyme behavior is very important in not only maintaining cellular homeostasis, but also in various aspects of practicing medicine, such as designing drugs or monitoring patient response to a stimuli. At this point, it is very important that you have a firm grasp on understanding the basics of enzyme kinetics and various enzyme mechanisms, as well as a good understanding of the Michaelis-Menten equation. From this point on, we will look at how inhibitors and allosteric controls are used to influence both Vmax, the rate at which product can be formed, and Km, the specificity of the enzyme to the substrate. So if you don't have a good understanding of Michaelis-Menten kinetics at this point, pause here, go back to Sean's video and/or the Khan Academy videos to review before proceeding. As always, here are our learning objectives for this section. Note that when I use the shorthand MM, I am indicating Michaelis-Menten, and when I say LB, I am indicating the Lineweaver Bruke plots. Again, be sure you truly understand and can fully answer these learning objectives as you prepare for exam 2. When we talk about enzyme inhibition, we must first define what is an inhibitor. Simply put, an inhibitor is a molecule that reduces an enzyme's activity either by reducing its affinity for its substrate or by reducing the rate at which it makes product, and sometimes an inhibitor can do both. There are two main classes of inhibitors. Inactivators, which bind permanently to the enzyme, are reversible. These are usually used by the cell to trigger degradation of the enzyme, as well as a means to control and possibly shut down a specific metabolic pathway. Reversible inhibitors, as the name would suggest, can decrease the enzyme's activity, but can be removed to allow the enzyme to resume normal activity. This is a common tool used by cells when they want to slow down or briefly stop a certain metabolic step that needs to be switched back on quickly. Therefore, the cell may not want to completely stop or degrade the enzyme. Often, reversible inhibitors will resemble the enzyme substrate structure such that it will bind but then never react or reacts very slowly. When we discuss reversible inhibitors, we can further classify these inhibitors as either competitive, uncompetitive, or non-competitive. Competitive inhibitors compete directly with the substrate by binding to the enzyme's active site. Often, these inhibitors can either be the product of the enzyme-substrate reaction itself, in which the product competes for the substrate and blocks further product formation. This is often called direct feedback inhibition. Another type of competitive inhibition is the transition state analog, in which a molecule that is very similar to the transition state of the reaction binds to the enzyme substrate active site and is non-reactive, thus preventing product formation. Uncompetitive inhibitors bind to the enzyme substrate complex, but not to the free enzyme. This binding distorts the active site to prevent product formation. Non-competitive or mixed inhibitors are able to bind to the free enzyme or the enzyme substrate complex in order to distort the active site and prevent product formation. Because inhibitors can affect either the enzyme specificity for the substrate or the rate at which we see product formation, the Michaelis-Menten kinetics of the enzymatic reaction shift in response to increasing inhibitor concentration. And these shifts are specific to the type of inhibitor. Therefore, when we analyze the enzymatic activity in the presence of an inhibitor, we can predict the type of inhibitor we have by looking at how the Lineweaver Bruke plots shift in response to increasing inhibitor. For instance, with competitive inhibitors, we will see an increase in the apparent Km. With uncompetitive inhibitors, we will see a decrease in the apparent Km as well as a decrease in Vmax. While mixed uncompetitive inhibitors show a decrease in the apparent Vmax with variable changes in the apparent Kmax of either increasing or decreasing. Let's look a little closer at each type of inhibitor. In competitive inhibition, the inhibitor binds to the free enzyme at the active site. Because of this, the apparent specificity of the substrate decreases, which translates to an increase in the apparent Km under Michaelis-Menten conditions. The Lineweaver Bruke plot of 1 over reaction velocity versus 1 over substrate concentration at increasing inhibitor concentration shows an increasing slope with a constant x-intercept and an increasing y-intercept. Because both the substrate and the inhibitor compete for the same active site in the enzyme, the effect of the inhibitor can be overcome if substrate concentrations are sufficiently increased. In uncompetitive inhibition, the inhibitor only binds to the enzyme substrate complex, therefore seemingly increasing the affinity of the enzyme for the substrate, but slowing the overall rate of the reaction. Under Michaelis-Menten conditions, this appears as a simultaneous decrease in Kmax and a decrease in Vmax. In the Lineweaver Bruke plots, the slopes remain constant. However, we see the y-intercept and the x-intercept both increase, the x-intercept becoming more negative, with the y-intercept becoming more positive. These types of inhibitors are especially good at very high substrate concentrations. Non-competitive inhibitors can bind either the free enzyme or the enzyme substrate complex, which appears as an overall decrease in product formation, but the apparent substrate affinity can either increase or decrease. However, because Vmax always decreases, the slope of the Lineweaver Bruke plots, which is Km over Vmax, will always increase in the presence of increasing inhibitor. Enzymes can also be controlled through bioavailability, in which the cell controls how much of the enzyme in a certain metabolic step is expressed or degraded in order to control the overall concentration of the enzyme in the cell. We can also control enzymes through allosteric effectors, which are molecules that interact and bind with an enzyme to increase its activity or affinity for the substrate. Covalent modifications, such as cleaving zymogens to obtain the active enzyme, or phosphorylation to turn on or turn off certain enzymes, can also be used to control enzymatic activity and therefore certain metabolic processes. Allosteric control of enzymes can also be positive. Activators are small molecules that bind to enzymes in order to promote affinity of the substrate, sort of like how we saw in the binding of oxygen to hemoglobin to promote the binding of more oxygens. In one example from the text, the binding of ATP to ATcase increases the Vmax and decreases the Km for the production of N-carbamylyl aspartate. Feedback inhibitors can either be direct products or products further down the metabolic pathway that can bind and decrease the enzyme's affinity for the substrate, therefore slowing down the entire metabolic pathway. For example, CTP, which is a product 6 steps after ATcase, can bind to ATcase, increasing the Km. However, because this is a type of competitive inhibition, if there is a buildup of aspartate, which is the substrate for ATcase, we can overcome this inhibition and still reach Vmax. As we begin investigating the regulation of various metabolic pathways such as glycolysis or gluconeogenesis, we see that the covalent modification of enzymes through phosphorylation or de-phosphorylation is often used. Typically, these types of modifications are themselves governed by other enzymes. Zymogens are inactive forms of enzymes that require the cleavage of another enzyme in order to become active. For example, chymotrypsin is cleaved first by trypsin, then it is self-cleaved to form the active enzyme chymotrypsin, a very important protease that we have already studied and will study in more depth later. In this video, we have covered the various classes of inhibitors, how their kinetics are changed in the presence of increasing inhibitors, and how we can use Lineweaver Bruke plots to predict the mechanism of inhibition. We also explored some various examples of allosteric control. As we continue through the term, we will begin to explore enzymatic control in more depth as we discuss specific metabolic processes and how they are regulated through enzymatic activation and inhibition. Right now, however, it is important that you master the various classes of inhibitors, be able to discuss why, not just how, their apparent Kms and or Vmax's change in increasing inhibitor concentration, and be able to look at Lineweaver Bruke plots and predict what inhibitor is being used that is described by the data. And also, you need to be able to explore and explain why the cell might utilize a competitive inhibitor versus a non-competitive or uncompetitive inhibitor to control a specific enzyme or metabolic step. I will see you guys in class. Have a great day.